Good evening, everyone. Today also we will take some class because the syllabus need to be covered is much more. Hence, it is I'm going to take the class. So, this class title is the genetic code and its relation to the animal genetics and breeding. Of course, we need to deal with different type of the animals, including the dog, cat, cattle, sheep, goat, pig, and many more animals are there. And because of this thing, we need to study what is the genetic code and what is its important in animal breeding. So, coming to the introduction class, so the genetic code and its relation to the animal genetics and breeding is required for establishment of the understanding of the common genetic traits, especially in case of animal genetics and breeding, and it is an in-depth exploration of the particular subject is concerned. Then introduction is concerned, the genetic code is the fundament, the foundational to all biological forms, and in the animal genetics and breeding, it allows for the manipulation of the traits such as growth rate, disease resistance, fertility, and milk production. And this presentation explores as the genetic codes, their role and its application in the animal genetics and breeding is concerned. And what's a genetic code? First of all, you should know what's this genetic code. This diagram very properly depicts the genetic code. So it's a universal phenomenon where in which determines the phenotypic and genotypic characteristic of any living being in this earth, including the most small virus to the very big mammal, that's the whale. And it is the genetic code is the usually it is red means it is universal in nature, and the genetic code is not completely random in structure. So we need to understand what are all the genetic codes and traits. And what's a genome? First of all, we have to understand what is a genome. Then a genome is an organism's complete set of genetic instructions. And each genome contains all of the information needed to build an organism and to allow it to grow and develop to the full extent and the length of its life. So the organisms are usually made up of the millions of cells. So we cannot count the zeros in front of the cells also, and with each other their own complete set of the instructions for being and sustaining that living thing. So this set of the instruction is known to our genome, and it is made up of the basic unit called as the deoxyribose nucleic acid or DNA. So in each cell in an organism, so starting from a skill cells to liver cell, brain cell, or including the stem cells, so they have got a very specific set of instructions. So then the genome to genes. So we can understand the instructions in our genome are made up of the deoxyribose nucleic acid, whereas within DNA a unique chemical code that guides to the organism's growth, development, and also maintaining the health strategies. And this code is determined by the order of four nucleotide base pairs, that's adenine, thymine, cytosine, and also the guanine. So these we can call as the genetic code. These are the basic material which are responsible for the genetic code. And DNA has a twisted double helix structure in the shape of a twisted thing, and a single strands of the DNA are coiled up to form the structures are called as the chromosomes. So a single strand, if it is becoming the double, then it is coiling is called as the chromosome. And in animals, the chromosomes are located in the nucleus of a cell. All the animals do have the chromosomes in the nucleus, not in the cytoplasm also. Then within the chromosomes, the sections of the DNA are organized into genes. Then genes control different characteristics such as the eye color and height and many more things. The milk yield, the meat quality, the growth rate, the reproductive rate, then also the disease resistant pattern and many more things, especially in case of a living being or the livestock, and the size of the egg, size of the body, then the production rate and the sustainability of the animals and the food conversion ratio, converting the food into the meal and also to the meat. All these are determined by the genes. And all living things have unique genome. So the complexity of the genes, multiple genes, do determine the genomes. Hence, it is one of the science, most advanced science, and we need to understand what's the gene and what's the genome and what's the genome sequence. So there is a very big project happened that's called as the human genome. So the scientists went for the discovery of the human genome, and of course they made 3.2 billion bases of the DNA. But other organisms have different genome size. So the 800 crore population in this earth do have the different genetic code, and every individual do have the different codes for the gene basis for that. And if printed out of 3.2 billion letters, so it is impossible. For example, it will fill a stack of paper bag books of 200 feet or 61-meter high and fill 200 telephone directories. Each contain the 500 pages. Then it takes a century to recite if we are recited one letter per second for 24 hours in a day. So it's such a big thing and extend 3,000 kilometers, about 1,864 miles approximately, the distance from London to Canary Island, Washington to Guatemala, or from New Delhi to Hanoi. So such a big thing is there, and it's very complex in nature, and the scientists have been trying so to do the genome sequencing of individual person, and what are all the advantages of this thing is to know the disease pattern, how to control the disease, genetically inherited diseases, and many more things are there. And there are, for example, deciphering the blueprint of a life of any person. For example, there are one codon and there are the number two start and stop codons are there. Codons are the one which direct the gene for particular multiplication and to synthesize the required type of the proteins. And the third one is the non-standard codex. Then the fourth is the application part of this particular thing. And we have the DNA and RNA. The difference of DNA and RNA is very, very less. Deoxyribonucleic to ribonucleic acid. Deoxy, one oxygen moiety is missing. So the process of this takes place from initiation of the transcription, then the elongation of the transcription, then termination of the transcription, and different types of the RNA are produced. For example, the messenger RNA and the different RNAs are there in the cell and sometimes the cytoplasm also. Hence, the RNA to protein. So, mRNA is very essential thing which code for a very specific type of the protein, and the genetic code is the one which determines which type of the proteins need to be synthesized, which is required by the body. Hence, the role of tRNA translation into the ribosome structure and function, then always it is leading to the post-translational modifications are always in that day. So the evolution of the life itself took place a long back, about 65 million years ago, and somebody said that it is more than that also. But the genetic code is made up of, usually starting from the beginning, it has not changed. Usually the adenine, thymine, guanine, and cytosine, same thing. Whereas in case of the RNA, it is uracil instead of methionine. Then the universal genetic code is shaped by all living organisms in this earth. Then to form simplest bacteria, you can also say not the bacteria, even with the virus also, there are DNA and RNA virus are there. So the genetic material even itself is enough to have a big disease, especially in case of the human beings are considered, like COVID-19, etc. You know very well how the virus evaluated as a new strain and tried to dewash the human life. And the genetic code is redundant, then meaning the multiple codons can code for the same amino acid, and a unique amino acid can be directed by a unique gene. And the fifth one is the genetic code is not entirely universal. We say that they are universal entirely, but it is there are some minor variations in the codon usage between the species. So the genetic code, and it has got a direct relation to that of the animal genetics and breed. So what's the genetic code? Once again, coming to the genetic code, then the DNA is a basic genetic material, what we say, and carries genetic information from cell to cell and generation to generation. So it is incorporated into the gametes or the sperm or ovum. Finally, it is transmitted from the offsprings to offspring through several generations, maybe throughout the evolution of the animal kind, or also the bacterial virus. They are also going to have a very specific genetic code. So the DNA is a director which controls, regulates, and determines the nature of proteins to be synthesized in a cell in a particular time. So the, or else it can be called as the master or the director. For example, in a cinema, everyone is going to act as per the director's order only, and he is the one who creates the situation, who creates the scene, and ultimately executes finally in the film. Just like this thing, the DNA is the director of the genetics or the inheritance or the life itself, what we can say. Then genetic code usually consists of a process of translation, means change, requires transfer of genetic information from a polymer to the nucleotides to a polymer of amino acids. So here the translation and the requires the transfer of genetic information in the form of adenine, thymine, guanine, and cytosine, whereas this is later converted to the RNA, adenine, uracil, guanine, and cytosine. Then the mRNA requires or it directs the DNA and other things to produce the required quantity of the protein and type of the protein, which is very much essential for carrying the genotypic character from one the animal to its offspring. So understanding the genetic code is, of course, we need to understand the basic thing, especially the genetic code is a set of rules what the body has created, or the body itself is autonomically or without any control of the external atmosphere. It has created certain rules. The animal has to be like this. The liver has to go to this extent. Then the brain should be of this extent. Then the skull should grow up to this age, etc. Then all these are predetermined, and the DNA and RNA sequences are there to synthesize the required proteins and also the required materials for building the life of an animal or human being or whatever the plant. Everything is controlled by a genetic code. Whereas the codons, they sequence of three nucleotides correspond to a specific amino acid. So, for example, adenine, thymine, and guanosine, etc., they are called as the codon. Three pairs of the nucleotides they form the codon. Then the code is nearly universal across the species, making it fundamental biological process. So, the genetic code is a very basic thing which codes or gives some signals to the cell through the different nucleotides, adenine, thymine, guanosine, and cytosine, so that the cells will produce the required genetic material in the form of the proteins. So each species has got its own genetic code, and the DNA and the chromosome, everything get changed. So there is no unique thing, and we cannot make the animal to divide off this particular thing. So here you can see that genetic code is a group of nucleotides specify one amino acid and is known as codon, and the relationship between the sequence of the amino acids in a polypeptide chain. Polypeptide chain, and it's also called as the genetic code. And it's a group of nucleotides. Usually it happens in such a way that the group of nucleotides which code, especially the polypeptides, or else we can understand like this: it is a genetic code is a group of nucleotides that specify one amino acid and is known as the codon, and the relationship between the sequence of amino acids in the polypeptide chain is called as the genetic code. And because of the specific genetic code, the phenotypic and genotypic characters, the breed, everything is dependent. And the genetic code here is a simple calculation is there, how it is going to be calculated, especially in case of the uh, animals. So the in the first chart, the especially the single code is based on the only one, and number of codons there are four. Whereas the double code, it's called as the singlet code. So the doublet code is containing number of the bases in codon are 2, then the codon is 4 into 4 is equal to 16 in number. Whereas the triplet code, it has got the 3 codons, and the number of total codons is equal to 4 into 4 into 4, that is 64 number of the genetic codes are there. So ultimately, the genetic codes they produce or they direct to produce the uh, specific type of the amino acids. Then, if we have the triplet code, then 4 into 4 into 4, 64. You can see the permutations and combination here from the box 1 to the last box also. So they are totally 64 genetic codes. Then they were going to produce 20 different amino acids are concerned. Hence, the process needs certain calculations and everything, and ultimately it need to be understood as ultimate aim of genetic code is to produce the amino acid which is required by the different cells. Then properties of the genetic code. Then the code is a triplet, means it is containing three nucleotides, and the codons code for amino acids, and three codons do not code for any amino acids. So So hence they function as stop codons. And when we are going to have the permutations and combination of adenine, thymine, guanine, cytosine, uracil, etc., there is one stage where three uracils are there, then they synthesize phenylalanine, and three guanosils are there, they are going to produce the glycine. And the code is the degenerate, second one. So in the triplet code, 64 codons are possible only, and each amino acid is coded by more than one codon. For example, the serine is coded by uracil, cytosine, uracil, and also uracil, cytosine, and cytosine. And similarly, the sequencing of these nucleotides will evolve in the different type of the basic amino acids. Then, what's a property of a genetic code means the code is non-overlapping. Means if a code is there, then there is no confusion between these codes, so that the unwanted amino acids will never be produced, because ultimate aim of this code should be always read in groups of three nucleotides from a fixed starting point. So there is no confusion, because whenever these three nucleotides are there in the permutation and combinations, then they will be directing the ribosomes to produce a very specific amino acid. So the code is a comma-less, means in between triplet codes, no letters are wasted. In triplet codes, no letters are wasted and continuously they are written. And no punctuations are needed between two codons. That's the basic rule of reading. Then the codon system, I mean, little bit detail. What's a codon system? That's the each codon, a sequence of three nucleotides will be there, especially for producing a very specific amino acid or a stop signal in the protein synthesis. Whereas the universal nature of the genetic code means that the same codon in almost all the organisms will correspond to the same amino acid. So adenosine, thymine, and guanosine. So it will be uniform in all the animals. They, if they are paired, then it will produce the same type of the produce irrespective of the species. That's what the codon system says. Then here you can see the DNA, a universal genetic code. Then it has got multiple components. So it is said that we hold the ancient text, one of the most ancient texts, and usually it is always predetermined, and it is the, suppose there is in a dog, if you, it will say it would be white if all evolved the same molecule separately, then there is no difference between any of the animals. All would have been similar, but it is not like that. That would be the separate cultures developing the same exact language independently. Then the DNA, what makes organisms different, and especially in case of the sequencing of the DNA, also known as the genes. So there is no difference between the DNA or genes or many types, they are used interchangeably. All organisms have it. So we must have forgotten from common ancestors. So our ancestors may be more than millions or millions here. They had also the similar type of the DNA structure, and during the evolution process, the changes like mutation, etc., they took place. Then the phenotypic and genotypic characters also change as per the time advances. Hence, here there are certain basic structures are there. You can see that, especially in case of the mammals, the dog, it's having 78 chromosomes and 90% DNA shared with that of the human being. So the 78 chromosomes are very nearly, it is matching to that of the human beings, and 2005 initially sequence. Then cat is having 38 pairs of the chromosomes, and it will not change from one another to another, and 90% of this will match with that of the human being. Then uh, 2007 initially they sequenced, whereas the elephant has got 56 chromosomes, and 70% of these chromosomes usually shared between the human, and the chimpanzees, they are called as the pan troglodytes. So they do contain very near number of the chromosomes, 48 and 98% similarity between that of a chimpanzee, then also the human beings. So accordingly, this goes who are our ancestors. Let us take some more knowledge from the reptiles. Always we see that the Komodo dragon is one of the creature. It has got 40 chromosomes and the countless base pairs. And in the 2019, the scientists they sequenced this type of the protein. So whereas the alligator, then sea turtle have got little similarity and the difference, as you know it very well already. You're all maybe experienced this thing. If you have not started, start reading. Then tortoises do contain the 52 pairs of the chromosome, and the base pairs are very high in this tortoises are there when compared to the base pairs which is present in the reptiles. Then it starts from the 15 billion may also be there. Whereas the fish, they are, for example, the zebrafish, on which we are always trying to do some these experiments, and there are certain similarities between the human beings. So, compared to the zebrafish, then figu is another uh, fish sometimes. It has also got 40 chromosomes. Then the whale shark or whale and shark are the different type of the things. Even then, the whale or the shark do contain 102 chromosome pairs in their body. So the 32 crores base pains do initially sequence, whereas in case of the electric jellyfish, and the scientists did not leave it. Once again, it do contain the 52 amino acids, and you need to help us. So it's okay, we'll see what best we can do. Then properties of the genetic code, usually there will be initiation codes, so codons are there, then also usually the termination codons are always need to be examined. Then the structure of DNA and RNA, always DNA, deoxyribose nucleic acid, or the ribose nucleic acid. They offered it to the applicant, and they may be under use also. So the RNA ribonucleic acid plays a very important role in encoding that the information for a protein synthesis. So DNA consists of varieties of the matter like nucleotide, base pairs will be there, then adenosine, uracil, then cytosine and guanosine. All these came, and one side the origina had also the same thing. DNA consists of nucleotide base pair, then like ATC, adenosine and etc. So while RNA causes A, U, C, and G. So the RNA or ribonucleic acid, it plays an important role in decoding that information for protein synthesis. And the DNA synthesis consists of the nucleotide A, T, C, G, etc. And we know it very well. And the DNA contains ATCG, adenosine, thymine, cytosine glycolysis, whereas the RNA is a single stranded. It contains, instead of thymine, it contains the uracil, and its function is to transfer the genetic information, whereas the DNA stores genetic information in it, whereas the RNA transmits it. And translation, decoding, and genetic information. Then translation is a process of converting mRNA sequence into the protein. And this involves three main steps: initiation, elongation, and termination. And each codon in the mRNA is matched with a tRNA molecule carrying the corresponding amino acid. Then the role of genetic code in animal genetics. So we have dealt out many things about the human genetics, but what is the role of this genetic code in animal genetics? It is the same as usual. In animal genetics, the genetic code plays a critical role in determining the physical and biological traits of an organism. Traits such as the disease resistance, fertility, and growth rate are determined by a specific type of the genes. And by understanding and decoding the genetic code, the animal breeders can select animals with desirable traits to improve breeding outcomes. Then the genetic variation in animal breeding is also another thing which is going to operate as the variation refers to the differences in genetic makeup among the individuals within a population. And these variation form foundation for selective breeding. And usually the animal breeders can capitalize these differences into produce the animals with the desired genetic material. For example, an animal with high milk production, high meat production, high wool production, and also the disease resistant genes. Then also the source of genetic variation includes the mutation, recombination, and gene flow. So these are the different technologies. And what are all the sources of the genetic variation are? Why there is a genetic variation? It is due to the mutation, a random changes in gene sequence that can result in new traits or traits. Whereas the second one is the recombination, means genetic reshuffling during the reproduction leads to new gene combinations. Whereas the third one is the gene flow and movement of the genes from one population to another through the breeding of migration. Whereas the other thing, where with the polygenic traits in animal breeding, so polygenic traits are controlled by multiple genes. Poly means multiple genes rather than a single gene. The examples include milk yield in cattle and body size in pigs. So these are controlled by polygenic traits in pigs and the cattle respectively. So the polygenic traits are influenced by both genetics and the environment and making them complex to manipulate through the breeding. So this is also the thing. Just like a human being with the genetic trait of intelligence, and it need to be supported by the environment like a good school and good coaching, good teaching, good learning material, good environment and good health. Then the breeders use the quantitative genetics to assess the contribution of these genes and apply selective breeding strategies accordingly. So the definitions, once again, we will sum up the definition like a gene is a sequence of DNA which codes that is the instructional manual for amino acid. So gene means always it is an amino acid, and these amino acids are building blocks of the protein, and proteins are very much required for the animal, and the animal has two copies of each gene which can vary, and the chromosome is a very large unit of the DNA containing many genes, thousands of the genes, and each chromosome is made up of pair of the chromatids, two pairs. Cattle have 30 chromosomes found in most cells of the body. Cattle cells, if you take it open, and if you count the chromosomes, the number of chromosome pairs is 33, 30 chromosomes. In sperm and eggs, chromatids exist as unpaired single entities. In chromatids and that's called as the haploids and diploids. Therefore, the animal inherits half of each chromosomes and thus one copy each gene from each parent, either mother or the parent. An allele, it's a form of version of a gene. So it's also nothing change. An animal can have the same allele on both chromosomes, that's called as the homozygous, or different alleles. For example, it is also called as heterozygous. And genes can have multiple alleles such as MCR1 responsible for the red coat color variants, however, MCR2 is most common in the animal. An animal can only inherit one allele from each parent, not than that of. So there is a restriction. And a recessive allele is one only has effect when an animal inherits this version of the gene from each parent. For example, it is always denoted by the small letters. The dominant alleles are always noted by the capital letters. So a dominant allele affects an animal when one or two versions are inherited, and most harmful alleles are recessive. Most harmful alleles are recessive, just like the cancer genes. And SNP means single nucleotide polymorphism is a single unit of the DNA which varies between animals within the same breed species. It is also called as single nucleotide polymorphism. A SNP may be located on a gene on a region between the genes on a chromosome. So SNPs, single nucleotide polymorphism genes are markers used to genotype of the animal and genomic selection. So a halotype is a group of the single nucleotide polymorphism located close to each other on the chromosomes, then which are usually inherited together. Then, what is a genetic code? Means, ultimately, to sum up all these things, it is a dictionary that corresponds with the sequence of the nucleotides. So, it is the term given by George Gamow. So, the genetic code is the set of rules by the information which is flown into DNA or RNA sequence. Then the discovery of the genetic code is made by the Watson and Crick, the famous Watson and Crick model. Then John Gamow, he also postulated that three letters code must be employed to encode 20 standard amino acids, which are required by the animals and also different type of the mammals. Then the genetic code is always made up of the four pairs of the nucleotides correspondent basically found in case of the DNA. So the collection of codons is called as genetic code. So such type of the codons are called as the genetic code. For 20 amino acids, there should be 20 codons, different codons. Here you can see that the chromosomes are located on the nucleus, and on the nucleus the DNA is present, and on the DNA the genes are present. And the codon should have three nucleotides to impart specificity to each amino acid for a specific codon. And one nucleotide, four combinations are there: 2, 16, 3, 64, 4 into 4 into 4, 64. whereas 4 into 4 it is 16. So it is going like this: adenine, thymine, guanine, cytosine, etc. So three of these nucleotide are going to code for one amino acid. Always this chain was like this thing, and the genetic code can be expressed in a simple table with 64 entities, and these 64 are going to give rise to 20 different amino acids. Then the genetic word is dictionary consists of the genetic words called as the codons. Genetic code is also called as the codon, and each codon consists three bases triplet. There are 64 codons and 61 codons code for 20 amino acids found in the protein, and three codons do not code for any amino acids, just like three uracids. So the types of the codons, there are called as the sense codon, single codons, start codons, stop codons. Then sense codon is the codon that code for the amino acids are called as the sense codons. And signal codon, these codons are the codons for the single during the protein synthesis and are called as single codons. For example, AUG, UAA, UAG, and UTA. So there are two types of the signal codons: terminating codon and initiating codon. So what's a terminating codon? Whenever the protein synthesis is started, then at certain stage we need to stop. So whenever the codons come like UAA, UAG, UGA are called as the termination codons. So always to stop the protein or amino acid sequence synthesis, this combination should come, and they are also called as the nonsense codons. It's very peculiar thing. So these are called as the nonsense codons. Then nonsense codons, and this type of the codons do always attention. The anticodons means they are acting in exactly opposite direction to that of the codon. So the sense of the tRNA which pairs with codon of the mRNA during translation is called as the anticodon. And tRNA anticodon loop is like this. So it is going to have the wobble position. 3 and 5 position are called as body position, and ultimately ending up with the different between the codon and anticodon. Always it is present in the codon present in the DNA and RNA, but anticodon is always present in only in RNA and never in DNA. Codons are written 5 to 3 direction, whereas the anticodons are in the opposite direction, like 3 to 5. So 3 to 5 and 5 to 3. So anticodon it is from 3 to 5, whereas the codons are from 5 to 3. Then the codons, anticodons of RNA molecules have to pair with more than one codon. Then codons are sequentially arranged in nucleic acid strand, while anticodons are discretely present in cells with amino acid attached or not. So here what we can say is codon defines which anticodon should come next, and the amino acid to create the protein strand, and anticodon helps in bringing a particular amino acid at its proper position during the translation process. So characteristics of a genetic codon, it is always a triplet code and commaless, non-overlapping, the coding dictionary, and it's also got a very specific degenerative code, universality of the code, and they are having the chain initiation codes are there, chain termination code, and we will see what are all these things. And genetic code is always it's a triplet. Here you can see that it is a triplet nucleotide base pair, and there are 64 codons are there. Then the universality means the genetic code is universal. AUG is the codon, which is for the methionine in the mitochondria, whereas the codon, same codon. That's the AUG codes for isoleucine in the cytoplasm. In the mitochondria, it is going to produce methionine, and also in the isoleucine in the cytoplasm. Whereas with some exceptions noted in genetic code is universal. Then non-ambiguous, the genetic code is non-ambiguous, and thus one codon cannot specify more than one amino. That's it. So the non-overlapping also, they will not overlap. One base cannot participate in the formation of more than one codon, and this means that the code is non-overlapping. Then there is a continuous translation process is there, and the gene is transcribed and translated continuously to form a fixed starting point and to a fixed stop point. And the punctuations are not present between the codons. So the code has polarity. The code has definite discretion for reading message, which is referred as the polarity. Then reading message from left to right and right to left will specify the different amino acids. For example, UUG stands for leucine, whereas from right to left it is GUU, which stands for valine. So the degeneracy of the genetic code, so it can be coded by more than one codon, and this is called as the degeneracy of the genetic code. So a specific amino acid need not be produced by unique combination. It can also be produced by the different type of the nucleotide combination. Then wobble hypothesis. So this Watson and Crick, among the Crick postulated wobble hypothesis to account for the degeneracy of genetic code. Then, according to this hypothesis, the first two bases of the codon pair according to the normal base pairing rules with the last two bases of the codon, anticodon. And base pairing at the third position is codon is a wobble. Just like you can see that here, wobble pairing is there, whereas GAG and CUC are the normal pairing, whereas GAG and the CUU are called as the wobble pairing. So the wobble hypothesis explains the degeneracy, and always it is going to express the existence of multiple codons for a single amino acid. Although there are 61 codons for our amino acids, the number of tRNA is far less, around 40, which is due to the wobbling hypothesis. Hope you can understand what is a wobble hypothesis and degeneracy and many more terminologies. Then the biological significance of degeneracy of the genetic code, because it has got a biological significance, means only 20 codons would design amino acids and 44 would lead to chain termination. And the probability of mutating to chain termination would therefore be much higher with non-degenerate code. And this is the flow of the genetic information. So from DNA, it undergoes the transcription process, then the genetic code will be changed from RNA to that of the translation, then the required type of the protein is synthesized or the peptides are formed by using different type of the nucleotide combination or the codons. Then the clinic significance means mutation can very well explain using the genetic code. So the point mutations. What is the point mutation? It is silent and then missense, then nonsense also. Then frame shift mutations are the clinic significance. What is a silent mutation means? A single nucleotide change from A to adenosine to guanosine. Guanine, same amino acids is incorporated. Mutation goes unnoticed here. So this is called as the silent mutation in this particular table you can see that particular effect. For example, ATG methionine, so GAA is glycine, alanine, and glycine. Or, for example, then later the same thing, the GAG may also produce the this GLU amino acid, and this is called as the silent mutation. Then messense mutation means single nucleotide chain A to C, different amino acid incorporated. Then loss of functional capacity of the codon. That's called as the missense mutation. Please note down all these important terminologies. What are all the missense mutation, etc. Then the nonsense mutation is defined as a single nucleotide chain from C to T, cytosine to thymine. Stop codon is generated in mRNA, which is represented by UAG, and a premature termination of the chain may be incomplete with that of the life, and this is called as the nonsense mutation. Here the examples are given, a flow chart, etc. So here replacement of single nucleotide is being occurring, and this is called as the nonsense mutation. And frame shift mutation, insertion of removal of the bases can alter the reading frame with resultant incorporation of different amino acids. So here you can see that the recessive defects, especially some of the genetic defects, then BLAD means bovine leukocyte adhesion deficiency syndrome, normally occurring protein needed to white blood cells or in nucleocytes, which are bodies the infection fighters. So they are not produced. So BLC is tested carrier is BLAD, bovine leukocyte adhesion deficiency. Then mule foot is another condition which is responsible for the toes of the foot are joined, giving a animal single hoof instead of the cloven hoof. That's called as the mule foot. Especially it is the characteristic of equine species. Then DUMPS means deficiency uridine monophosphate synthase on or many enzymes contributing to the normal metabolic process. Then CVM means complex vertebral malformation. This causes stillborn calves, then abortions and early embryonic losses in case of the animal. And factor S1 is blood clotting disorders may also be there, and these are all the genetic codes and expression codes. And CIT is citrullinemia, means accumulation of the ammonia and other toxins in the blood of young calves. It is called as a genetic disorder. Then another condition is called as brachyspina, is a abortion and stillbirth, shortened spinal cord, long legs, and abnormal organs are there. Then CD is direct test or the this cholesterol deficiency syndrome, and cows have reduced or no cholesterol, resulting in death of the cows, especially the young, as the cholesterol is very much required for the body to protect. So these are all the recessive defects, and it is very difficult for identifying, and it is very dangerous. Of course, the dominant genes are not so problematic, whereas the recessive defects are going to cause lot of genetic oriented diseases. Whereas we can see here in the next slide the hold status of the animal. It's also called as the current indirect test, and the gene responsible is also called as the indirect test and test through hold. Gene and expression codes are written on the right side table, whereas the red alleles are the coat colors in animals. For example, red gene MCR1 is there, whereas the red variety is red variant gene is there. Whereas the black red gene MCR1 once again, then black gene is considered uh, the same thing, and genetic codes are transmitted. For example, here there is a very clear picture how a cell and the DNA, then also the codons, etc., are there. So here this is the chromosome where the different base pairs are present in case of humans and other thing where cat contain 30. So the chromosomes, chromo means color. In the center there are centrioles are there. Then chromatids are the arms of these chromosomes. Maybe the two pairs are there. So they are always present inside the nucleus. Then the chromosomes will be present in the nucleus. Then a part of this strand of the chromosome is, if you cut it and magnify, then that is called as the DNA. Whereas the SNP, single nucleotide polypeptides, outside the gene are also present and no effect on the protein production and function. So the different type of non-coding SNPs are also present. Then coding SNPs are going to code the proteins required. Then meal proteins, like we are telling the A1 and A2, so it is also related to the genetics, and the beta casein is also called as A2 protein, beta casein. So the gene responsible for this thing is CSN2, a homozygous for A1 type of the alleles, heterozygous for A1 and A2, then homozygous for A2. So now earlier this concept was very much spread, like this A2 milk is very good for the health and A1 is deleterious to the health of the human beings, but there are no proofs at all to prove that the A1 is dangerous and A2 is very beneficial. Uh, so these are all the some trade competitions which are created by some group of the people who always fight for the benefit. Then kappa casein is also known as CSNO2. So beta casein and kappa casein. So all these are going to change the milk protein traits. But nowadays, already it is proved that almost all the crossbred cattle do have the A2. The buffalo is having 100% A2, and all the local cattle breeders do have the dominant A2 or beta casein. So we cannot go and take this as the defending our local cattle because it presented the crossbred cattle and also in case of the buffaloes. Then there are a lot of possibilities and limitations. There are ethical concerns are there to change the nucleotide sequence and getting a new breed or disease resistant breed, high yielding, etc. Whereas it is also applicable in case of agriculture. Different type of the plant varieties can be evolved out just like the BT cotton and the brinjal, etc. But lot of opposition came and they have almost stopped it. Whereas the personalized medicine can also be another possibility of the genetic coding, whereas the data privacy is also a challenging thing. Suppose this is available to a lab just like the mobile number, whether the it can be sold to the external person or not. So the data privacy is always a threat for this particular uh, coding. Then finally, we will come to the conclusion that the genetic code plays a very important role, especially in case of the animal's traits, and is central to modern animal breeding practices. So, with advancement in genetic technologies, breeders have more control over the enhancing the desirable. breeds, especially the desirable things, and the genetic characteristics are always the challenging matters, especially in case of the breeders, to get evolved a new breed, new cattle breed, and the same thing which holds also holds good to the human races are concerned. However, there are a lot of ethical constraints are there. Then one must guide the application of genetic manipulation to ensure the responsible and sustainable development in the field of animal genetics. So these are all the things, and we need to conclude that the genetic code is one of the important thing in the life of the animals, and at the same time which determines the different type of the things, and also it is responsible for evolution of the different breeds of the animal, and also the beneficial for the human generations are concerned. But at the same time, lot of ethical constraints are there. They should not be misused. And at the same time, the persons who are handling all these things should always maintain the confidentiality among the people, and the data should be kept very, very confidential. And if you understand the proper things, especially about the genetic code, etc., we can develop different strains, etc., for the benefit of the humankind, and at the same time, not causing a dangerous or threat to the nature by incorporating the gene of our own intention. So we'll see. We have to balance all these things, and with this, I thank you all for staying with me for the about 1 hour, and thanks a lot. Bye. See you tomorrow.